Your watch tells you what time it is, but what if it instead told you about the timing of the events of your life? This watch might tell you about your birth, your 16th birthday, your college graduation, your wedding day, the birth of your first child, and so on. This watch still tells time, we're just looking at it a bit differently. This is exactly what a molecular clock does because it measures the number of changes in a genome over time. These changes, or mutations, accumulate over time, and most of these changes occur at a constant rate. This allows us to see both how individual species evolve as well as when species branch off from each other evolutionarily. Molecular clocks are calibrated against evolutionary events that are known from the fossil record. But from these known events and rates of genomic change, we can estimate relative dates of evolutionary events that aren't already documented in the fossil record. For example, if you know that a change occurs every 700,000 years and you can identify 42 changes overall, then you have determined that the two species diverged about 600,000 years ago. Molecular clock analysis can be used to create evolutionary timelines for organisms that don't have much of a footprint in the fossil record. Many plants, fungi, and soft-bodied animals don't leave behind fossils because they don't have the hard body parts to do so. Molecular clocks to the rescue. For these guys, we can use the molecular clocks of organisms that do leave behind fossils and make inferences about their genomic changes based on those that have given us a good record. Molecular clocks can be useful not only for identifying when living organisms diverged over time, but also viruses such as HIV. Working backwards using a molecular clock, scientists have been able to identify when HIV first jumped to humans, which was back in the 1930s. Molecular clocks can help organize an evolutionary timeline as well. By comparing genomic data of different organisms, scientists can identify how organisms are related and where they fall in the evolutionary tree of life. For example, they have learned that bats and dolphins are actually more closely related than sharks and tunas. Pretty amazing, huh? Even your Rolex isn't perfect, and neither is the molecular clock. Though most changes occur at a constant rate, the rate of change may vary in different genes, at different times, and among different organisms. In certain instances, this can be accounted for by calibrating the molecular clock to multiple genes instead of just a few, but it still poses a problem for scientists using molecular clocks. Even when calibrated, estimates from molecular clocks can be problematic because our fossil record only goes back about 550 million years ago. However, molecular clocks are used to date evolutionary events that occurred over 1 billion years ago and even farther back than that. Scientists assume that the clocks have been constant for that entire time, but that's a big assumption. Molecular clocks keep track of a different sort of time by measuring the number of changes in a genome over time instead of how many seconds, minutes, or hours have gone by. From identifying dates of divergence to filling in gaps in the fossil record to simply organizing events into an evolutionary family tree, when calibrated with known events from the fossil record, molecular clocks can be useful for a number of different evolutionary analyses. While useful, molecular clocks are not foolproof. Rates of change may not be constant, may not be the same in all organisms, and may be different for different genes. But as the science and technology improve, so will our understanding of molecular clocks and how they tell evolutionary time.